We are ready to start the final session of the evening. And this is Jean-Marc Lemaitre, who will tell us about reprogramming, I think.
First, I have to thank Martin and Alex for this invitation, and also for this very wonderful meeting. So for many years in my lab, we are interested in the cell plasticity, in development and aging. And we developed some reprogramming strategy to try to fight aging. And everybody now beneficiates of this famous hallmark of aging, initially 9, but now other. And what we know is that we can follow aging with this hallmark, and for sure they are involved they are at the root of the age-related pathology. In the lab, we focused on cellular senescence and epigenetic alteration. Why? Simply because of my simple vision of what occurs in the cell when they stress. Cell experience during their life many different stress, and stress generates damage. The cell has to repair the damage, and if she can repair the damage, she is able to continue to do the job. But we know that there is some hallmark, and we can observe what we call an epigenetic drift, with a transcription modification and an impaired cell plasticity. If she's not able to repair, we know that she can trigger apoptosis or senescence with this specific SASP, very deleterious for the cell. So early on, we asked the question, is cell aging reversible? We decided to start from scratch. And we beneficiate from this famous discovery of Yamanaka with these 4 factors. He showed that we are able to convert an adult cell's skin fibroblast initially into a pluripotent stem cell, raising the cell identity, and then redifferentiation of these cells in any kind of cells open the door to the cell therapy. So we decided to use this cocktail, and we started from age cell and senescent cell, the 2 types of cell we guess to be present in tissue with the age, and we tried to reprogram them, and it didn't work at all with this cocktail. And some years after, 6 papers published, and one from Professor Yamanaka too, saying that cellular senescence and aging are a break and a barrier to reprogramming. So because we were resilient, we have to be in aging, and we decided to try another cocktail published the same year by James Thompson, replacing Calais-4 and Semic by Nanog and Lin28, with the same efficiency of reprogramming on adult cell, but it did not work at all. And we decided to mix the factors, and with the 6th factor, we were able to derive IPS efficiently from senescent cell and from centenarian cell. And when we redifferentiate them, we observe the fully rejuvenated physiology. We reset the gene expression profile, we elongate the telomere, we reset the proliferative capacity, even from senescent cell. Mitochondria physiology was reset, and the cells regain the ability to go back into replicative senescence. Even more, we compare the efficiency of reprogramming with 6 and 4 factor, and we observe that we are more close to human embryonic stem cell with the 6 and with the 4 of Yamanaka. So then we explore the opportunity of this amazing technology, and with this technology, for sure, we can of IPS with patients with disease. And we develop IPS with premature aging syndrome, like QLE case. We spoke about that during this meeting. And also with, for example, mesenchymal stem cell from osteoarthritic patient, and to model the pathology after redifferentiation and to recapitulate the pathology to eventually screen for molecules to prevent the disorder. But we were also involved in a cell therapy proof of concept from the degenerative intervertebral disc, and to make the proof of concept of the usage of IPS to differentiate into progenitor of the cartilage and to be able to regenerate the intervertebral disc. But in parallel, we have another question. Is it necessary to go to pluripotency to rejuvenate an aging cell? And we decided to develop in vitro and in vivo model to show this. And we developed mouse model, one reprogramming mouse model with one allele of the reprogramming cassette, and another model with the progerin, the one copy of the gene forming the progerin. So initially we start from skin fibers from this model, and contrary to what was shown before with the reprogramming into IPS, we decided to induce the expression of gene a short period of time, and we observed that at this time we decreased the DNA damage, contrary to what was observed with the fully reprogramming process that generates stress. And we also decrease the senescence. We also stimulate the autophagy, but we did not switch the metabolism as it was described for the fully reprogramming into IPS. Then we decided to analyze gene expression by RNA-seq with these two models induced into OSCAM expression, and we identified 395 genes in common, differentially expressed by the induction of the 4 factors early on. But what was really interesting is that we start with fibroblasts. So effectively, when we look at gene pathway triggered, we see wound healing, epidermis development, for example. But interestingly, we see also kidney development, osteoblast differentiation. So many different functions of different tissue. So we decided to look at what occurs in vivo in this model. And initially, we tried to reproduce the publication of Ocampo in 2016 from the lab of Giuseppe Belmonte, and he used a similar mouse model, but an homozygous progeria, so living only 5-6 months, and we use an heterozygote progeria, so living 2 times more and more close to physiological aging. And we use this chronic induction. So today the week during the life of the animal, and we observed a similar increase in lifespan of our model if we only induce 2 days a week during the life of the animal. Then, because 2 days a week with 1 milligram per meal of dogs was highly expressing the cassette, we decided to decrease the level of induction, but not chronically, but during all the life of the animal continuously. And we observe similar effect on the lifespan extension. Then we would like to know what occur with only one reprogramming period. So we decided to trigger reprogramming during 2.5 weeks, but early in the life of the animal to see what occurs. With 0.2 milligram per minute of dogs, nothing occurs, but if we increase a little bit at 0.5, we observe that there is an increase of lifespan of the animal in old age. So we induce at 2 months, and we have an increase in the lifespan late in the life. So we decided to look at what occurred at 8 months, close to the end of the life. But what we observe, what was absolutely amazing, is that we change immediately the body composition of the animal and during all the life. So in fact, we maintain the muscle mass, and we decrease the increase of fat with the age, but during all the life of the animal and only for a short induction early in the life. And it's this modification of the body composition is associated to an improvement of fitness, as measured by this rotarout test and the mid-holding time on the wheel, and also the grip test. They increase really the fitness, the strength of the muscle. Then we decided to look at age-related pathology, and we started with the osteoarthritis. And what we observed is that treatment early in the life, 2.5 weeks, at 8 months of the animal, we see that there is an increased cartilage volume, there is a decreased surface degradation, and the connection with the subchondral bone was also involved, and we observed that there is an increased bone volume and also bone thickness of this part of the knee. And if we look at osteoporosis on the tibia cortical region, we have also an increase of the bone volume, and due to an increased bone mineral density on the cortical regions. Then we look at the structure of the tissue, of different tissue. We observe that, for example, in the kidney, there is an improvement of the microarchitecture of the kidney. When the mice age, there is an increase in size of the glomeruli, and also an increase fractional mesangial area. But we observe that with the early induction, we preserve the structure of the glomeruli and the surface area. And similarly, in the spleen, with the age, there is a sort of distortion observed between the two parts, the white pulp and the red pulp. This is the marginal zone, and it's possible to evaluate this and with a score, and we observe that we maintain the distortion, we preserve, the distortion of the separation between the two parts. And Alex said yesterday that fibrosis is aging, so we looked at fibrosis, and the early treatment prevent fibrosis in kidney, in spleen, and also in lung, and there is a tent for the liver and the heart. But the last tissue, and not the least, the skin, and it's absolutely amazing because you can observe this is the same magnification. So definitely we observe that all the layer of the skin are improved late in life with the early treatment of reprogramming. And even more, if we shave the mice, there is an improvement of the hair recovery by an early treatment of 2.5 weeks. So because of the distal effect, we guess that probably epigenetic mechanism has to be involved. And what we decided to do, and not to develop clock, because we were on the progeria model, and for other reasons that we will find out later, we start to identify this epigenetic drift by a methylome analysis on Illumina beads with the 250,000 CPG reference, and we identified the differential methylation site related to aging that correspond to this epigenetic drift. And then among these sites, we look at what is differentially, what sites are differentially methylated early on after the treatment, and the consequence at 8 months. And what we observe is that at 8 months, when we treat early on, there is a differential methylation site among the aging differentiated methylated site, but it's specific for each organ. So you understand it's complicated to have a clock in this condition, but anyway, what was absolutely amazing is that almost 100% of this site differentially methylated at 8 months among after the treatment were reversed in methylation. So the methylation status is the same that after the induction. But what was more amazing is when we compare to the site targeted early on, the methylation targeted early on, there is no overlap. So it means that the reprogramming initiates an epigenetic mechanism, but this on methylation site, but it's not maintained. It just propagated. And at the end, the site differentially methylated at 8 months, they recover a yolkful epigenetic information. So finally, we observe that this early induction of reprogramming is organ-specific, and it's initiated by transient OS-KM expression, for sure, 2.5 weeks at 0.5. It's not maintained, but propagated. It restores youthful epigenetic information on the aging DMS, the DNA methylation site, but it also increased longevity. It improved body composition during all the life of the animal. It also improved the tissue ultrastructure. It prevents fibrosis and also some age-related disease. So we believe that it's a new opportunity probably to treat and to prevent aging. So this last slide to acknowledge all the people of the lab, especially Olivier Milavé, Quentin Allais, Nora Lebon, and Paul Bensanoul, really involved in this project, and other who left the lab, but also involved in this project, and also all the platform and the financial support we have. We have also a postdoc position to work on reprogramming. So thank you.
Thank you so much, Jean-Marc. We have a couple questions.
Thank you very much. Very nice presentation. You showed this progerian model, but do you have any data on wild type? And also, how can you separate what is from the partial reprogramming and what may be an effect from doxycycline itself?
Yes, when we do with doxycycline itself, there is no effect, and you can see also with the modification of the metabolism we observed this morning with this doxycycline, we see that on mitochondria we have no modification, no change at all, and we also did experiment on wild type mice. And this is the result. You know that there is also an increase in lifespan, and these mice live 2 times longer, but we also treat at 2 months of the life of the animal, and we observe an effect late in the life.
Very cool. Pekka?
That was very nice, thank you. So your doxes, of course, are taking effect on every cell. So my question, I guess, then is, what do you contribute the effects on? Is it on an cellular level of every cell being reprogrammed to a level? But on the other hand, your new data on the epigenetic clocks maybe is suggesting that it would be an effect on some type of a progenitor or a stem cell pool that then propagates further down with time. How do you see this?
For sure, the cassette is present in all the cells. When we reprogram typically all the cells, but for sure we don't know if there is some cells with more efficient action on the tissue. But and we didn't develop a single cell analysis, so each time we look at global on the tissue, but we observe that there is a modification in microarchitecture and so on. So we believe that there is a global tissue effect.
Right. Thank you so much, Jean-Marc.
Thank you.
And